Lab 11 project. I'm Riley Martin. I'm going to go over the background. First, we're going to go over respiratory volumes and capacities. First definition is the tidal volume, the volume of air breathed in and out without conscious effort. Next is the inspiratory respiratory volume. This is the additional volume of air that can be inhaled with a maximum effort after a normal inhalation. And the expiratory respiratory volume, which is the additional volume of air that can be forcibly exhaled after a normal exhalation. So basically, those are when you take a deep breath in and a deep breath out. And residual volume is the volume of air remaining in the lungs after maximum inhalation. And the vital capacity is the total volume of air that can be exhaled after maximum inhalation. Total lung capacity is the amount of air with the vital capacity and the residual volume. So that's every part of the lung, how much air that could be filled. The next is the anatomical dead space. This is the volume of air in the conductive division of the respiratory system during one respiratory cycle. And the alveolar ventilation rate. And this is a measure of the body's ability to dispose of carbon dioxide and deliver oxygen to the tissues. Next, we're going to talk about what occurs during physical activity. Well, as physical activity increases, the depth and rate of respiration will increase because the sympathetic nervous system initiates bronchodilation, which increases respiratory and alveolar ventilation. Why? Well, the body needs more oxygen supply energy for cellular respiration and aid in the production of ATP. These modifications will allow for more strenuous exercise. So basically, the body needs more oxygen so that it can have enough energy to exercise continually. But what happens to CO2? Well, the carbon dioxide is removed at a quicker rate to match the increased oxygen consumption. Next, what happens in the brain during respiration? Well, there are three types of brainstem respiratory centers where respiratory cycle is controlled. First is the ventral respiratory group. This sets the basic respiratory rhythm. The primary respiratory muscles receive stimulations from the VRG, and the VRG is located in the medulla oblongata. Another part that is also located in the medulla oblongata is the dorsal respiratory group. This receives information about the conditions in the body and communicates with the VRG to modify the respiratory rate. As pH levels lower, which means it will become more acidic, the CO2 will increase. This will cause the chemoreceptors in the DRG to signal the chemoreceptors in the VRG to make adjustments to the respiratory cycle. Pontine respiratory group is our third one, which is located in the pons, the mediator of information from the cerebral cortex, hypothalamus, and the limbic system to the DRG, the synapses with the VRG to further modify breathing. All these respiratory centers are made to maintain homeostasis in our lungs. This is just an image showing where we can find the VRG, DRG, and PRG. Kind of gives a better understanding of where they are located and their functions.
Hypothesis, independent and dependent. If the subject's physical activity increases, then the alveolar rate will increase. The independent variable is physical activity. The dependent variable is alveolar rate. Procedure, experimental design, spirometry. Spirometry is a test that measures the volume and flow of air inhaled and exhaled out of the lungs. This is measured with a spirometer, a device that captures and records the rate and depth of breathing. The spirometer or spirometry can be used to detect diseases like asthma and chronic obstructive pulmonary disease, COPD. With spirometry, we can measure the following volumes and capacities: respiratory volumes and respiratory capacities. Respiratory volumes are tidal volume, TV, the volume inhaled and exhaled during one cycle of breathing. The normal range is 500 milliliters or 0.5 liters. Inspiratory reserve volume, IRV, is the excess of air, TV, that is inhaled with maximum effort after normal respiration. The normal range is 3,000 milliliters or 3 liters. Expiratory reserve volume, ERV, the excess of air, of the tidal volume or TV, that is exhaled with maximum effort after normal respiration. Normal range is 1,200 milliliters or 1.2 liters. Residual volume, RV, is the air remaining in the lungs after maximum exhalation, 1,300 milliliters or 1.3 liters. Respiratory capacities. Vital capacity, or VC, is the total amount of air that is inhaled and exhaled, 4,700 milliliters or 4.7 liters. Inspiratory capacity, IC, the maximum air that is inhaled after a normal expiration of 3,500 milliliters or 3.5 liters. Functional residual capacity, FRC, is the amount of air remaining in the lungs after normal expiration. Is 2,500 milliliters or 2.5 liters. The total lung capacity, or TLC, is the total amount of air that the lungs can hold, 6,000 milliliters or 6 liters. Procedure, experimental design, control, and experimental group. The control group is the subject sitting at rest, will breathe into the spirometer, taking normal, calm breaths. The experimental group is the subject running in place post-exercise and then breathing into the spirometer.
The data we collected from this experiment was the tidal volume, which is the amount of air breathed in and out without conscious effort, the respiratory rate, which is the amount of breaths taken in 1 minute. This is calculated by dividing 60 by the amount of time taken to have one respiration. The minute respiratory volume, which is the volume of air breathed in 1 minute, which is calculated by multiplying the tidal volume by the respiratory rate. The anatomical dead space, which is the air in the conductive division of the respiratory system during one respiratory cycle. And we know that at rest, this is 150 milliliters, and during exercise, it doubles to 300 milliliters. And the alveolar ventilation rate, which is a measure of the body's ability to dispose of carbon dioxide and deliver oxygen to the tissues. This is calculated by doing the tidal volume minus the anatomical dead space divided by the respiratory rates. The tidal volume at rest was 0.9 liters, and post-exercise it was 2.18 liters. The breaths per minute, or respiratory rate, was 16 at rest and 24 post-exercise. The minute respiratory volume was 14.3 liters at rest and 52.67 liters after exercise. And the alveolar ventilation rate was 12 liters at rest and 45 liters post-exercise.
So today we'll be going over the results and observations for Lab 11. And it'll be about the formulas for percent change and the outcomes for those. So the formula for percent change is percent change equals post-exercise data minus at rest divided by at rest, and then you multiply that by 100%. The tidal volume was the first piece of data that we did this for, and it was in liters, so it was 2 liters minus 1 liter divided by 1 times 100. So that was a 100% change because it was doubled. And then respiratory rate came out to 24 minus 16 divided by 16 times 100, which was 56% change. Minute respiratory volume was liters divided by minutes, and that was 53 minus 14 divided by 14 times 100, which came out to be 279% change. The anatomical dead space was measured in milliliters, and it was 300 minus 150 divided by 150 times 100, which came out to be a 100% change. The physiology related to its change. So the anatomical dead space represents the air in the conducting zone that's unable to exchange gases within the blood, or with the blood. So at rest, the parasympathetic nervous system decreases airflow, which minimizes the anatomical dead space. which is why it's at 150 milliliters. And then during exercise, the sympathetic nervous system dilates the airway that increases airflow, and that increases that amount of air that's unable to exchange, increases the ADS, which is why it's doubled at 300 milliliters. The increased airflow compensates for the increased ADS, and gas exchange increases as needed to maintain pH homeostasis and energy production. And then last but not least, the alveolar ventilation rate was measured in milliliters per minute, and that would be 45 minus 12 divided by 12 times 100, which comes out to be 275% change.
So to conclude, we have our conclusion questions. Our first one is compare the tidal volume and respiratory rate changes from experimental data to the background information. Does the data match general observations? Compared from post to pre-exercise, the change in tidal volume was 122% and change in respiratory rate was 56%. So the data does match general observations that it would increase with the rate of breathing also increasing. And our second question is discuss the percentage change in AVR rate and the physiologic effect it has on meeting the metabolic needs of the body. The rate change in AVR was 275%, which had an increase. AVR is important because it measures the body's ability to dispose of CO2 and deliver oxygen to the tissues, meeting the metabolic needs of the body. And our third question is to discuss the hypothesis and if the results support or reject the hypothesis. And our hypothesis states if the subject's rate of physical activity increases, then the AVR would increase too, supporting the hypothesis that shows in the data.